Hi everyone, welcome to Subhash Chandra Tutorials. I am Subhash Chandra Shabannavar. Today I am going to discuss the topic molecular basis of inheritance, where we are going to study what is nucleic acid, what is nucleoside and nucleotide, what are the components of nucleotide, and what are the types of nucleic acid. Let's start the class with definition of nucleic acid. Nucleic acid is a polymer of nucleotides, where nucleotides are the monomers which were made up of three components. The three components are pentose sugar, that is 5-carbon sugar molecule, and nitrogen base, they are purines and pyrimidines, and phosphoric acid. Nucleic acids are of mainly two types. They are DNA and RNA, that is deoxyribonucleic acid and ribose nucleic acid. Components of nucleotide are pentose sugar, nitrogen bases, they are purines and pyrimidines, and phosphoric acid. Start with pentose sugar. Pentose sugar are of mainly two types. They are ribose sugar and deoxyribose sugar. In RNA, ribose sugar is present, and in DNA, deoxyribose sugar is present. It lacks oxygen molecule. Let's study the structure of both ribose sugar and deoxyribose sugar. Both sugar molecules are 5-carbon compounds. Both contains furanose ring. Furanose ring contains 1 oxygen and 4 carbon, and 5th carbon of a sugar is present as in the form of arm. Let's number the carbon atoms that are present in ribose sugar. This is carbon number 1, carbon number 2, carbon number 3 and 4. 5th carbon atom is present in the form of arm. Carbon number 1 is attached to hydrogen and hydroxyl group. Similarly, carbon number 2 is also attached to hydrogen and hydroxyl group. Same as in case of carbon 3 also. In case of carbon number 4, hydrogen group is attached, and in case of carbon number 5, CH2OH group is present. The main difference between ribose sugar and deoxyribose sugar is present at the position, that is carbon number 2. Now we can see structure of deoxyribose sugar. Deoxyribose sugar is exactly similar to ribose sugar, only the difference is present at carbon 2, that is hydroxyl group is replaced by simple hydrogen. The second component of nucleotide is nitrogen base. Nitrogen bases are of two types. They are purines and pyrimidines. Purines are of further two types. They are adenine and guanine. Pyrimidines are of three types. They are cytosine, thymine and uracil. Nucleic acid contains 2 purines and 2 pyrimidines. In case of DNA, 2 purines, they are adenine and guanine, are present, and in case of pyrimidines, cytosine and thymine are present in DNA. In RNA, purines, they are adenine and guanine are present, and pyrimidines, the thymine is replaced by uracil in case of pyrimidines in RNA. Purines are dicyclic structures or compounds, while pyrimidines are monocyclic structures. 4 nitrogen atoms are present in purines, which are located at 1st, 3rd, 7th and 9th position in dicyclic ring. In case of pyrimidines, 2 nitrogen atoms are present, which are located at 1st and 3rd position in dicyclic ring. The glycosidic bond that forms between pentose sugar or ribose sugar takes place between 1st carbon of pentose sugar and 9th position of purine. At 9th position, we can see nitrogen atom in case of purines, and glycosidic bond formation takes place between pentose sugar and pyrimidines at first carbon of pentose sugar or ribose sugar or deoxyribose sugar and first position of pyrimidine. At first position of pyrimidine we can see a nitrogen atom. Third component of nucleotide is phosphoric acid. Chemical formula of phosphoric acid is H3PO3. This is the structure of phosphoric acid, where we can see oxygen get attached at one end and other three sides OH groups are present. Phosphoric acid is get attached to the pentose sugar at fifth carbon of pentose sugar, and the bond formed between phosphoric acid and pentose sugar is called as phosphodiester bond. Now we are going to discuss or study how these three components are going to unite to form a nucleotide. This is the pentose sugar. This is carbon number 1, carbon number 2, carbon number 3, carbon number 4 and carbon number 5 is present in the form of arm. Purines or pyrimidines are get attached to first carbon of pentose sugar, that is nitrogen base. If it is purine, the ninth position of purine make glycosidic bond with the first position of pentose sugar or ribose sugar or deoxyribose sugar. If it is pyrimidine, the first position of pyrimidine going to make glycosidic bond with the pentose sugar. Phosphoric acid is going to get attached to fifth carbon of ribose sugar, that is phosphoric acid here we need to study how phosphodiester bond is going to form. That's why I am going to write this OH group here. Phosphodiester bond that forms between phosphoric acid and the ribose sugar or a pentose sugar is going to form by eliminating water molecule from this OH and this OH group. Now the phosphodiester bond is formed between phosphoric acid and the pentose sugar. Glycosidic bond is formed between the nitrogen base and the pentose sugar. Here the pentose sugar is ribose in case of RNA and deoxyribose in case of DNA. The structure except the phosphoric acid, that is the pentose sugar and the nitrogen base, going to form the glycosidic bond and it is going to be called as nucleoside. If the phosphoric acid is get attached at fifth carbon of a pentose sugar by making phosphodiester bond, then the whole structure is called as nucleotide. Here I am taking DNA as an example, where deoxyribose sugar is going to get attached to the nitrogen base, that is adenine, that forms a nucleoside. In particular, this is deoxyribose adenosine. And then the phosphoric acid get attached by formation of phosphodiester bond with deoxyribose sugar. Then this is called as nucleotide. In particular, deoxyribose adenosine monophosphate. In next class, I am going to discuss structure of DNA and types of DNA. Please share and subscribe to our channel to get instant notification of every video that we upload in our channel. Thank you.